Embryological development of the pleural and pericardial cavities involves intricate cellular differentiation and migration patterns, structures, and molecular mechanisms. We will explore clinical implications, repair strategies, in this enlightening journey through embryology. During embryonic development, the mesoderm plays a vital role in the formation of the pleural and pericardial cavities. The mesodermal cells undergo a process of differentiation and migration to contribute to the development of these structures. Initially, in the early stages of embryogenesis, the paraxial mesoderm gives rise to somatic cells, which will later play a significant role in pleural and pericardial structure formation. Somites, which are segmented bilaterally along the embryonic axis, give rise to a variety of important structures, including the vertebrae, ribs, and musculature. Within the somites, specific mesenchymal cells, called sclerotomy cells, are responsible for the formation of the pleural and pericardial cavities. These cells migrate ventrally and contribute to the development of the pleuro-pericardial membranes, which establish the separation between the pleural and pericardial cavities. The folding process during embryonic development is crucial for the establishment of the pleuro-pericardial membranes. Various molecular signaling pathways, such as the TGF-beta pathway, BMP signaling, and WNT signaling, play intricate roles in regulating this folding process. They control the proliferation, differentiation, and migration of the mesenchymal cells, ultimately leading to the fusion and formation of the pleuro-pericardial membranes. The septum transversum, which originates from endodermal and mesodermal cells, plays a crucial role in the proper positioning of the heart and formation of the pericardial sac. It contributes to the formation of the diaphragm, which acts as a barrier between the thoracic and abdominal cavities. Additionally, the septum transversum provides crucial signaling molecules that regulate the development of the pleural and pericardial cavities. The formation of the pleuroperitoneal fold is a critical step in the establishment of the separation between the pleural and peritoneal cavities. This fold arises from the lateral plate mesoderm and grows downwards, creating a barrier between these two cavities. It ensures that there is no communication between the thoracic and abdominal cavities, allowing for the proper development and functioning of these distinct regions. The respiratory diverticulum, an outgrowth of the foregut endoderm, gives rise to the lungs. During development, it interacts with various surrounding structures, such as the foregut endoderm, splanchnic mesoderm, and pharyngeal arches. These interactions are essential for the proliferation and differentiation of the lung bud, ultimately leading to the formation of the pleural structures. Neural crest cells, originating from the neural tube, migrate extensively during embryonic development and contribute to various structures within the body, including those involved in pericardial cavity septation and cardiac septum development. These cells play pivotal roles in the formation of the membranous portion of the interventricular septum and the separation of the atria and ventricles in the heart. Multiple molecular signaling pathways, such as the hedgehog, notch, and WNT signaling pathways, are involved in the regulation of pleural and pericardiac cavity development. These intricate signaling networks control cell proliferation, migration, and differentiation, ensuring the proper formation and organization of these essential cavities. Disruptions in the development of the pleural and pericardial cavities can lead to various clinical consequences. One common manifestation is the accumulation of fluid within these cavities, resulting in pleural and pericardial effusions. These effusions can impede organ function, cause respiratory distress, and even lead to cardiac abnormalities, highlighting the importance of understanding the embryological basis of these conditions. Abnormalities in the development of the pleural and pericardial cavities can result in a range of congenital anomalies. These anomalies include congenital diaphragmatic hernia, CDH, wherein a defect in the diaphragm allows abdominal organs to protrude into the chest cavity. Pericardial cysts, fluid-filled sacs within the pericardial cavity, and pleural adhesions, abnormal attachments between the pleura and surrounding structures, are also among the clinical consequences of disrupted development. Transcription factors and growth factors play pivotal roles in the molecular regulation of pleural and pericardial cavity development. Transcription factors such as NKX2.5, GATA4, and TBX5 are critical for the proper differentiation and specification of mesodermal cells involved in these structures. Growth factors, including FGF, BMP, and VEGF, provide essential signaling cues that regulate cell behavior, proliferation, and differentiation during development. Tissue engineering holds great promise for the repair and regeneration of pleural and pericardial structures. Researchers are exploring various strategies, including the use of biodegradable scaffolds, stem cells, growth factors, and gene therapy, to promote tissue regeneration and restore normal structure and function in individuals with congenital anomalies or acquired defects. In summary, the intricate development of pleural and pericardial cavities encompasses various complex processes, including cell differentiation, migration, and molecular signaling. Understanding these mechanisms is vital for comprehending clinical implications and developing innovative strategies for repair and regeneration. Ongoing research in embryology aims to improve patient outcomes and expand our knowledge of the remarkable transition from embryonic development to functional organ formation.